Okay, yeah, got it. Okay. So yeah, thanks for this invite. I thought that, last time that I was talking to some of you, there was indeed quite a lot of bird talk in my talk, and today you will hear about smaller organisms and how we model them. And I'm basically somebody who likes to have different approaches in my lab. So not everybody works on the same methodologies. And I thought that conceptually, I'm not talking about kind of like, particular mathematical methods here, but kind of like different kinds of ways of thinking about how to do theory in terms of how broadly you approach each question. So one aim that we're doing is that we are investigating some sort of abstract idea where people say that, yeah, surely some process should favor something should happen. And this is something where intuition can sometimes be unreliable. And then we are trying to build models so that we don't really investigate any particular species there. That's why this organism that I'm depicting there is the sort of crazy fanciful one. But we are investigating the validity of a sort of chain of reasoning that people have in their heads about what evolution should be favoring. And the other one is then, what I also like doing equally much is to collaborate with somebody who actually knows their particular organism really well. And this is a picture of Daphnia. This is clearly a female Daphnia because there's a lot of eggs developing inside her. It's kind of interesting that they have their eggs kind of like towards the backside, not in the front, like a mammal would be doing. And you will see how these specific systems, they can actually teach us quite a lot about the sort of big things like bed hedging happens to be a concept that I'll be talking about when I come to the Daphnia. So, but I start with the first thing here, and this is really work by Cora Klein, who is a PhD student in my lab. She's in her final year now, and you can see that she's looking at an insect. And her approach, the first time I heard about her existence was that she sent me an email, which basically said that, she's still an undergrad and she would like to do a master's thesis in my lab. And very unusually for a student, she already had a very clear idea of what she wants to do. And I read that email and I felt like, oh yeah, there seems to be something in there. But you always worry as a supervisor, like, maybe this has been worked out already. So I found some papers that are relevant, and it turned out that she had actually found those papers, but she felt like, she has actually good reasons to think her idea is different. And what she wanted to do is to, here comes the crazy organism here. If you're familiar with how these sort of inheritance diagrams work, you realize that the round ones are the females, the square ones are the males. And this is some sort of a weird thing that she's creating here. Like the life cycle doesn't totally make sense. Like there's two females coming together and somehow they are producing a bunch of males, or you have two males coming together and without the help of any female, they are producing a bunch of females. So why would you ever want to construct mathematical models of such reproduction? The reason, and I'll come back slowly towards this sort of craziest of all the examples that she wanted to do, is that the reason is that we have quite good theory in population genetics about how sexually antagonistic selection works, so that there can be alleles that have positive effect on both males and females. This is the yellow dot here. Or they might be, both effects might be negative. So you see the blue one, or it might be somewhere kind of where it's antagonistic, so that it has a positive effect on male reproductive success, but a negative effect on female reproductive success. And it's quite clear that, if both of these effects are negative, like in the blue one here, then it's not good news for such an allele. It should actually, we make the prediction that it should disappear from a population if it has arisen via mutation. If it's a strongly positive one, then it's quite clear it should invade and fix. If it's only very weakly positive, then there might be stochastic problems of achieving this, but basically we get the straightforward prediction that this is good and that this evolution and selection should favor this. It's a bit more complicated for these cases where it's good for one sex, but bad for the other. And for mammals, we have a pretty straightforward prediction. This is not our work. This is earlier work that, the positive effect on one sex just has to override the effect on the other sex. And then you will have a net benefit because each allele spends on average half of its time in males and half of its time in females. So this is fairly straightforward maths. So we get the invasion, no invasion threshold like here. But there's also a sort of slightly less straightforward result that is still mathematically quite simple to derive, is that there's this differently shaped area, which is about whether this allele will, after it has invaded the population, will it also reach fixation? And if it's inside this orange area, it will do so. And in between, we have this green area where what are the rules actually here? You can see that the allele will invade, but it will not fix. That means that we have conditions where there can be a stable polymorphism, that some individuals have it and others have the ancestral, the so-called wild type allele. So this is old stuff. And I put this picture of some sort of random mammal there because this assumes like every individual has one mother and one father and all these standard things that we often take for granted if we are human beings. And if you're, by the way, wondering, kind of like talking about humans, is there actually evidence that there's such alleles? There's actually lots. So this is not my work. This is just people, these three people who looked at this fantastic data set, the UK Biobank, and looked at whether there's evidence that humans have things in their genome that can have such different effects on males and females, and they actually found a lot. If I just look at the last line of the abstract there, evidence for sex differential selection is particularly strong for variants affecting reproductive success. But it gets a lot crazier if you go to something like insects. And this is an example. It's called the crazy ant. And it's actually not called crazy because its genetics is so crazy, but because their movements are sort of particularly crazy if you disturb the nest. But this is relatively new research where it was found that almost every male in this population has an allele that, if it's homozygous in females, so that the females have two copies of the same thing, it kills the females. And most females in turn carry one allele that is lethal in males, which means that when they create males, half of them die, which is super wasteful, but still this kind of thing has arisen and seems to be stably maintained. And this means that, when these really strong effects can be found in insects, and particularly haplodiploids, and I'll explain to those of you who can't remember what haplodiploid means, what it is. We sort of have to rethink how do these benefits and costs in two different sexes actually play out mathematically. So what you see here is the way inheritance works in haplodiploids. So once again, the round ones are the females and the square ones are the males. And there's always, for these chromosomes that are these colorful things here, females are diploid, just like humans. We have two chromosomes each, including the X chromosome, which is XX if you're a female. But males in haplodiploids are haploid, so they have only one set of chromosomes. So the way reproduction works, if you're an ant or a wasp or a bee or something like that, is that the fertilized eggs develop into females. You can see that, there's always the male contribution is in the females, but the males are produced by the mother alone, by the queen alone. So males have no father whatsoever. And this has all kinds of consequences. If you are, for example, a deleterious allele that has negative effects on the organism, it will always be seen by selection if it's residing in a male. So recessivity doesn't even have a meaning here because they cannot hide behind another copy of the same gene, whereas they can hide if they're in females. There's also the thing that works over generations, that alleles are actually more often found in females than in males. If you sample a random allele at a random point in time, just because females just have more genes in them because everything is duplicated, it might be, you could argue that maybe from a gene side perspective, it's more important to be good for the female than for the male. And then there's this interesting thing that I already mentioned, that the male actually never has a father. So these three effects have been verbally talked about in the literature. And interestingly, each of them has been seen as the kind of driving force behind the results that exist for knowing how the sexual conflict can play out in these kind of insects. So from this picture, which was true for something like a mammal, if we go to haplodiploids, we have to decide on, what happens on the kind of like recessive versus dominant ideas about the effects of the alleles. The dominance factor, it can be set to 0, which means that we are looking at a completely recessive situation. And if we are moving away from the mammals and going to something like a wasp, we actually get a completely different set of curves here. And you can see that, previously for the mammals, for example, you could not have any allele invading if it was in the far left here, because it's kind of like too bad in its effect for the females. But here we are completely in the green zone, meaning that even if an allele kills homozygous queens, it can still spread if it's a tiny, tiny bit beneficial to males. And this is where Cora was placing her master thesis. She said that, okay, there are these results. I mean, the pictures that you saw below, they were already known in the literature. But she felt that the intuitive verbal explanations of why exactly these regions have the shapes that they have, there might be some more work to be done, because there are these three distinct verbal explanations of trying to provide understanding intuitively of what happens if you switch from diploidy to haplodiploidy. And she was saying that she thinks that the maths is actually correct, but the verbal explanations given might actually not be so clear as they could be. And for the sake of terminology, she actually switched from this, a male has no father. We just decided to call it the alternation effect, because it basically means that there's no direct way for a male to produce a male except going one generation via a female, so that alleles always sort of keep alternating between the sexes, more than just sort of being randomly chosen from the previous generation to becoming a gene in the next generation. So she pointed out that it's actually interesting when you look at the real diversity of reproduction in nature, because these effects are actually not always co-occurring. They always necessarily co-occur if you are something like a wasp, but there are other insects, there are other weird examples out there. And for those cases where she would like to switch on one of these effects at a time, or maybe two at a time, she was thinking, maybe if we just invent a life cycle that cancels one of these effects, but retains another effect, then we could actually see what is actually responsible for what. And just to show one example that has unusual features about them and that really exists, it's from this paper in the pea aphid. You have probably seen this organism if you have, like, you're trying to grow some plants. Sometimes you get these aphids on them. And you can see that the title of the paper is Masculinization of the X chromosome in the pea aphid. So what's going on here? If we look at the aphid life cycle, they are creatures that can reproduce both asexually and sexually. And the males are formed in a different way from how they're formed in wasps, or, well, also different way from how they're formed in humans or things like that. Because you can see that the male, just as an X chromosome, the XO terminology means that there's nothing else. It's just the X, there's no Y chromosome, meaning that when sperm cells are formed, they have always an X chromosome in there, just like the sexual females forms her gametes, the egg cells or the ova. And this A here, that just means autosomes. That's all the other chromosomes that are not sex chromosomes. And when you think about this system, then you realize that this actually gives us an opportunity to look at just one of the effects on its own, which is the fact that males, for any genes that are on the X chromosome, they are actually haploid. So there's nothing else, no other copy in there. Whereas, for example, the fact that alleles would spend more time in females than in males is actually no longer true, because if you look at how these the next generation is formed, there's always one thing coming from the mother and another X coming from the father. So there's much more balance in there. So this was just one example how you can tease apart these effects by investigating the consequences of very different life cycles and asking, how would a female beneficial and male detrimental allele, or vice versa, spread in each of these systems? And then just to make sort of a complete disclosure of everybody who worked on the team, because I was kind of like busily running around reorganizing my life and all that. We got Hanna Kokko joining the team, who is also a mathematically oriented biologist. I asked her actually to send a picture of her because I didn't have any, and she sent me this one. She actually does have a summer phenotype as well, so this is how to recognize her if you see her somewhere. So we three then worked through all these examples that we wanted to make these effects occur independently of each other. So I already showed you what happens in the sort of boring mammalian case, where none of these asymmetries play any role. And this is something that we denote with these symbols, where this hexagonal pattern that basically means that this sort of offspring can be either sex, it can be either male or female. But we wanted all the other combinations as well. So we were thinking, what could be a situation where the fact that alleles are more often in females than in males, that would operate without males being haploid and without the alternation effect between the generations. And this is what Cora came up with. So this is a situation where, once again, the offspring can be of either sex, but in this case, they might have a father, but the female is also capable of cloning herself. And if the ratios of this type of offspring, so that there would always be 2 of these for every 4 of those, if that is true, then these effects are as they are listed here, so that the alleles spend exactly twice as much time in females than in males, but none of the other effects apply because males are diploid and there's no particular alternation. Facultative sex, by the way, is quite common in nature. It doesn't always follow exactly the rules that were specified here with these exact ratios, but it is quite interesting to reflect that sexual conflict in facultative sex organisms would work in a different way than in, for example, wasps. So then the next task was to create a situation where the male haploidy would operate alone from the other effects. And I already gave you the idea. So the P aphid, or particularly the X chromosomes of the P aphid, if we had sexually antagonistic variation on that chromosome, it would work exactly that way. And this is just how it's depicted. Then we need the alternation effect operating on its own. And this is where these crazy non-existent life cycles come into play. So if we look at this, we have females only ever producing males, and males, if this was physically possible for them to actually produce females without any females being involved in providing the eggs, then just mathematically you would have an organism where the alternation effect operates, but none of the other effects do. It doesn't exist in nature, but we can totally investigate what would happen if it did exist. But then we also wanted to have cases where 2 of these effects co-occur without the third one being present. And this is a particularly interesting one, I think. This is also something that does not exist in nature, but this would combine all the necessary ingredients in the way we want. And this, if you look at the life cycle here, you see that this is almost like haplodiploidy. So once again, the males have only 1 set of chromosomes and the females have 2. But now it's not true that males never have a father, because sometimes this chromosome is just inherited from the father and nothing comes from the mother. And we call this the shuffle model because it's formed by, effectively, you take these chromosomes in the parental generation like a pack of cards, and you just shuffle them, and then sometimes you take one card and sometimes you take two cards, and then you decide that those who got two cards, they develop as females, and those who got just one card, they develop as males. Then a real case for change, we wanted to find a case where the first and the third effect operate, but the second one is excluded. And this is quite funny that it exists. If you have ever had head lice, then you have had an organism on your head that does something quite exotic called paternal genome elimination. So here, the males as well as the females are diploid, but in a funny way, the males always inherit one set of chromosomes from dad and one set of chromosomes from the mother, and they express that, but they never make that paternally derived chromosome to go to the next generation. So whatever the male has inherited from his father, it's kind of like a dead end for that chromosome to go to the next generation. And it's a bit complicated to think through, but it actually does satisfy all these requirements that the alternation effect works. The males are not haploid because they actually use two sets of chromosomes to express whatever their body is like. But the first one here also applies. Then finally, there's the requirement that we wanted to get the haploidy operating and the alternation effect operating, but without the first asymmetry here. And this we call it's a bit like sort of plant model. If you're a botanist, you know that field talks about the alternation of generations, where there's sort of sporophytes and gametophytes, and it's all very complicated if you're not a botanist. But effectively, it's a bit like, you know, two haploid things coming together to form a diploid thing, and one diploid thing being able to split into two haploid things. And in plants, this is not the definition of males and females, but we are saying that if it was, then it would satisfy all these criteria that we want to have for this particular case. So it's a bit like, you know, if we had antagonistic effects in plants that favor the sporophytes or favor the gametophytes, it would follow exactly the same math structure as if it was a sexually antagonistic allele in this particular funky way. And finally, we have the last row here where all the effects operate together. And this is something that always operates in all these wasps and bees and things like that. And this is what it looks like. You have already seen this. So that was kind of like quite a lot of different life cycles to think about. All of these, we can create the equations for the, you know, you already saw the lines or the curves that are the thresholds for when an allele can invade or when it can fix. So what you see here, it's always the sort of the small a for the males and small a for the females. This is the fitness effect that it has, this novel allele has if it's expressed in males or females. And sometimes you can see this age here. This is the dominance, so that it means that if you are diploid and you have your heterozygote, you have one allele of the wild type and you have one of this new type, what is actually the fitness effect of this combination? And you can see that the equations, they are sometimes, they have this age and sometimes they don't have this age, and it's a bit sort of hard to immediately see everything what is going on here. So I'll just show you a few examples of what can happen in the model.
May I ask a quick question, Hanna?
Yeah, sure.
I was just curious, does this list that you have here exhaust all the possibilities in nature, or are there other things?
Well, nature has other things. I mean, this is exhaustive in the sense that if you look at the kind of like left column here, kind of like if you want to have just look at all these effects, then it exhausts the combinations that these cannot occur in. But if you know, for example, here in Mainz, there's an ant researcher who just had a science paper on ants where males are actually chimeras, meaning that different cells in their body can have different genes having come from different parents. And that's of course kind of like completely outside, this world that we are thinking here. So it's complete in a sense of wanting to address this particular question, but nature always seems to be more creative about, how exactly you can create offspring and all that. So in that sense, it's not. If that answers your question.
Thank you. Yes.
Yeah. Okay.
I have a related question. Sorry.
Sure. Yeah.
So this categorization, these three effects, do you think that there's something like very fundamental about these three things operating? So the categorization to what extent is this a natural thing to do?
Well, it's a natural thing to do in the sense, if I can actually show it with this figure here that I'm just showing, that if we are interested in moving from the sort of normal diploid organisms, and this walrus just is my funny depiction of a normal one. And it's only normal in the sense that we are very familiar with it because humans belong to this category. And then we move to this other very, very common type of organizing reproduction, which is the haplodiploid insects. I mean, they're actually biomass-wise, people have been estimating that ants are just so unbelievably common on this planet that it's actually a very significant part of the world's biomass is in that sort of reproduction. Then these are all the effects that happen inheritance-wise when we move from this to that. But if we are thinking about, how do the PE aphids, for example, organize their reproduction, then we actually did a little trick there, which is that we didn't want to think about the complications that come from all these asexual generations between the sexual ones. So we just sort of shortened the life cycle to the essential differences. So we were interested in everything that happens between this and that simply because they are very, very common cases. And we use these alternatives to chart the effects of each of the three things that happen there. Okay. So
yes, sure.
Because there's 8 different scenarios here, and of course, I mean, you can pairwise look at all of them in very many different ways. I will just show you one example from the paper where we showed what happens. So you remember that there were these thresholds when things can happen on one side and when things don't happen on the other side. And here, this is just sort of 4 of the 8 cases depicted next to each other. The rest you can look at in the figure. It will take a bit too long to go through each of them, and it gets a bit boring when you're sort of explaining every single change that there is. But you can already see here that there's sometimes you get straight lines and sometimes you get these curved lines. And if you stare at this sort of thing long enough, you can actually produce a verbal summary of how the absence or presence of certain effects impacts the solutions, because they always have a sort of very systematic effect of where they shift the solutions towards. So this effect that alleles are more often found in females than males, whenever that effect is present, then intuitively enough, it does promote female beneficial alleles. So they need a smaller positive fitness effect on the female side in order to invade and to fix if this effect is there helping them. And that is true both for the invasion stage and for the fixation, the ultimate fixation that may happen. Whereas the fact that males are haploid whenever they are in a life cycle, this, again, quite intuitively, it promotes male beneficial alleles because they are never hidden behind another copy that might also be seen by selection. But also it makes the area larger where this polymorphism can happen. So remember, it was in the first slide, it was the green area where something can invade, but it cannot reach fixation. So these regions get larger if males are haploid. And this alternation effect, it's a bit of a more complicated beast because it's always harming the initial invasion of an allele that has sexually antagonistic effects. And this is irrespective of whether this allele is good for the females or if it's good for the males instead. But if this harming effect is not strong enough, so that if invasion can get going, it actually promotes the ultimate fixation. So it's bad for the maintenance of a polymorphism. So what I find quite pleasing about this work is, well, two things. One is that it was really, the whole idea came from a very young student who had never published anything before, and she was sort of willing to work through this really kind of, very, very systematic way, and getting quite a bit of insight on the way. But I think, you might not want to think about weird insects all your life, but what I think is very fundamental modeling-wise in this question is that often when we do the maths and we do it correctly, and then we think through kind of like, how to verbally explain what's going on in there. In this case, a lot of people have just sort of looked at the outcomes and then they think, oh, there's this one thing that is different between this set of assumptions and that set of assumptions. But the first thing that comes to mind, for example, the fact that alleles are more often in females than in males, it might actually not be responsible for the effects that you're discussing, or they might be responsible just for partially the change that you're observing when the maths moves from this set of assumptions to that one, because there's also other things happening simultaneously between these two organisms. And sometimes you have to do quite a bit more work before you can figure out, what precisely is responsible for what. And that was quite satisfying. But I promised that I will actually talk about two very different things here. And I will now move to the different story where this was not something where we got analytical results for, but I still think that it was very satisfying to work on understanding a system. And the reason why I'm not going to show you any sort of analytical results is that it really does get quite complicated when you think about the world of Daphnia, which are these little water fleas that you can find in ponds, freshwater ponds in most places on this planet. And this is a collaboration with Dieter Ebert, who has been working on this for a long time. And I already showed you what the creature looks like. It's something kind of like, several of them fit on my fingernails. And they are facultatively sexual organisms. So what you can see here is a female who is most likely developing a clutch of daughters. I can tell because she has quite a lot of eggs in there, and the majority of these egg clutches are daughters only. Amazingly, she can also produce a clutch of sons, and she does it less often than producing a clutch of daughters, but she looks very much the same when she's doing this. And this is quite amazing because sons are actually clones of their mother, and they are not haploid or anything like that. They actually have exactly the same genome as their mother. There's nothing like sex chromosomes in this system. How she makes them male, the mechanism is a bit unknown, but it's probably just sort of flooding them with some sort of hormone while they're developing, and then they get to be males.
Hanna,
Yes.
sorry, can I ask a clarification
Sure.
question?
Yeah.
So there are no sex chromosomes, but you distinguish between males and females?
Yeah.
How does that
So
work?
the way we distinguish between them is, so the definition of a male in biology is that you produce sperm. So a male would never be observed producing eggs like this, like the mother does. Sperm are very, very different sized than eggs. And once you have, maybe it's easiest to explain with the sexual reproduction stage, this is again a mother. She has produced a much smaller clutch of much larger eggs than in this case, and they need fertilizing. So she needs to meet a male, and then everything works just like it works in humans. So sperm swim to the egg, the sperm are tiny and the eggs are large, and then you get the two genomes and you get a fertilized young, who actually in this case always develops as a female. So sons are only produced clonally, whereas daughters can be produced either clonally, so that they are identical to their mother, or sexually, like this. Was that clear now?
Yes.
Yeah, it works. Okay. So here again, we sort of check the validity of intuitive reasoning. And now we also check a particular metaphor, which is that of bed hedging. So here you see a bed hedger who has sort of come with, I guess this is from some sort of financial website, because there's a pound symbol in here, where, you know, if your boat wasn't really good enough to keep you going, then you still have some other way of surviving. And bed hedging is something where, if we just sort of talk about how people use the term linguistically, it is apparently to do something in a situation so that if the situation fails or has a bad outcome, you will not fail or suffer as much as if you had not hedged your bets. There's sort of two metaphorical sayings that both sort of seem to relate to bed hedging when it comes to giving life advice. One is that you don't put all your eggs in one basket. And you can already see how I'm thinking about Daphnia here, because they sometimes do this kind of eggs and sometimes some other kind of eggs. But there's also this idiom that a bird in the hand is worth two in the bush. And they both actually seem to relate to certain mathematical ideas about bed hedging, because bed hedging also has a mathematical definition in the evolutionary biology literature. But if you think about these metaphors, they seem to suggest doing two completely different things. One is that you should diversify. You should maybe lay your eggs in completely different baskets, so that if one of them kind of, you know, a predator comes and eats your eggs, then the other one is still safe. Whereas the other metaphor, it's kind of like, well, if you already are holding one bird in your hand, then maybe you should do that and not try to go for the stochastic option where, you know, you go for the really big bird, but if you try to catch that one, then you might end up with nothing at all. So this sort of verbal intuition gets a little bit confusing immediately when you start think about it. But both can actually be forms of bed hedging, because the bed hedging definition in evolutionary biology is any strategy that reduces the variance in fitness at the expense of lower arithmetic mean fitness. So you produce on average fewer young, but you also produce them sort of more reliably when it comes to the distribution of the number of young that you manage to create. So people have looked at this population genetically for a long time, and this is one of the classic results. Here you see the means and you see the variances. There's sort of type 1 and type 2, and N is population size. It's a finite population, and the fixation criterion is basically, you can see that there's a trade-off between the mean and the variance, and if this quantity is larger than that quantity, then type 1 was actually more fit based on the bed hedging ideas, even if its mean offspring production might be lower than the other types. There's also another formulation where you're explicitly sort of saying that, okay, you have this trait that decreases both the mean and the variance of the offspring distribution, and then well, it's basically just sort of reformulating what we saw above in a slightly different way, so that the increase is favored if this trait causes a decrease in the mean scaled variance in offspring number that is larger than a factor, roughly population size, times the decrease it causes in relative mean offspring number. Whether that's more intuitive than the first one, I don't actually know, but it's sort of rephrasing it. So the reason why I was telling you about two different metaphors and why they both can refer to bed hedging is that we need to distinguish between the variance reduction in fitness, the actual number of offspring that you are producing, and the variance in phenotypes. What do the organisms actually look like? Because either increasing or decreasing the variance in phenotypes can lead to a decreased variance in fitness. And I'll just explain what I mean here. So in evolutionary biology, we talk about a diversifying bed hedger, which is something like a Daphnia mother sometimes producing this kind of offspring, and some other time produces that other kind of offspring, and maybe even a third type of offspring. And they actually do 3 different types of offspring, because they can produce asexually produced daughters, asexually produced sons, or these small clutches of eggs that need fertilization. But there's also conservative bed hedger idea in the literature where you don't go for these sort of different kinds that with the idea that, you know, hopefully one of them is kind of okay in these particular circumstances, but instead you converge on a particular phenotype that is never particularly good, but it's never complete failure either. And there's quite a lot of literature, for example, in plants, how the different germination strategies, are you a dry year specialist, or are you a wet year specialist, or should you produce something that is kind of like doing a little bit of okay, no matter how much it rains, or should you instead diversify by making some seeds germinate very, very slowly so that they only come out in a year when it has already rained quite a lot. Or maybe some of them are much more eager germinators, so that the first drop of rain, they already come out. So that's then a diversifying strategy. So these are kind of like how I think that they roughly correspond to the different kind of metaphors that we are told about. But back to the Daphnia. So this was a real study in the sense that we actually did go out in nature, and I told you already that they live in these sort of little ponds. This is the open sea, but these rock pools, this is a picture from southern Finland. They're sort of tiny little places where water accumulates, and that's where you find a lot of the Daphnia. And we recreated the world for the Daphnia by placing these buckets outside. So it, you know, natural rain could come and splashes of water could come and all that. And we seeded these populations by putting a particular number of males and females onto, and maybe sometimes no males at all, because the females can also do things on their own, and just seeing how they react to the environment where they are now. So this is just to remind you what the organism looks like. We did this in these beautiful islands where you can see that there's quite a lot of variation in how big the islands are and how big the water bodies are, and whether they are very exposed to sun, and the ponds also freeze earlier in the autumn when they are sort of exposed like this than when they're on the bigger islands. So the question really here is, how does a Daphnia female decide, in quotes, what to do next? Because she can produce males if she wants to. She can flip back and forth between a clutch of male eggs and a clutch of female eggs, or maybe these small clutches of fertilization requiring eggs. And the observation is that when it's still spring and early summer, they go through these asexual generations, just females. There's no males in the system at all. But then at some point, they switch to this more complicated setting. So late summer and early autumn, at least some of the females do this. They might do the asexual route, but this time they create males, or they go to the route of producing eggs that need to be fertilized. So then they need to find a male who has already been produced, most likely by somebody else. And then they fertilize the eggs. And these fertilized eggs, they are the only form of Daphnia that can survive the winter. They are packaged in these, they look like little pea pods, small and black, and they can withstand the winter. They can also withstand drying out. And the next season, after winter, it is all recreated from this route alone. So then you will get the females who then can start to go through this cycle again. And you can see that I already put some terms here, like S for the probability of doing sex, as in going and producing these sexual fertilization requiring eggs. If you don't do S, then you do 1 minus S, but then you can split that into the probability of going for males. So this is 1 minus S times M is the probability of male production. Or if you don't go for sex and you don't go for the males, then you are producing daughters asexually. And the question is, how do we figure out the optimal values for S and M throughout the season? So one summer can fit in a lot of Daphnia generations. So this is a fun modeling exercise. It's also very fun for the experiments, because this whole allocation problem, it's really complicated not only because the female has 3 options, female offspring, male offspring, or the sexual eggs, but also because she doesn't have perfect ability to predict what's going to happen. So one thing that is certain, because this is Finland after all, is that the ponds will freeze at some point. And if your lineage has not produced the sexually produced eggs by that time, it's like game over. Your entire lineage is dead because no adult can survive these circumstances. So therefore, female production should stop at some point, this directly produced females. But on the other hand, it's really good in the short term, because if you fill the ponds with your clones early in the term, then you have many, many, many more of those who can have sex late in the season, and you fill the pond's bottom with lots and lots and lots of eggs at the end. So, but then it's also complicated because winter is not the only danger, because sometimes even in Finland, it can be so warm that especially the smallest rock pools, they can dry out completely. And this is also something that can only be withstood by the sexually produced eggs in these little pods. So a lot depends on the weather. So we're trying to figure out the optimal S and M for each time point when there's this incredible environmental uncertainty at all the times. And we sort of tried to figure out how to do this analytically, and because the sort of genes through the males and genes through the females and all that, it got very complicated. So let's just sort of list why it became so complicated. Direct production of the females, it is paying off a lot if you have the time to have turned this lineage into sexual eggs by the time the season ends. But you don't know when the season is ending. Also, male production is really good if they have somebody to mate with, but males cannot force females to go to the sexual route. They can only fertilize those eggs that somebody has already prepared to become fertilizable. So therefore, the value of producing males depends on how many females there are and how many of them have decided that now I'm going to go through a sexual cycle. But also, if a lot of other individuals have been producing males, then if you're adding males to that pool, then they face a lot of tough competition, because any egg can, of course, be fertilized only by one sperm. So male production, when there's already a lot of males, can be a bad investment. Also, it takes about 2 weeks' time for each male to mature. So if you measure the male density now, you produce males and they will compete in the world that is 2 weeks from now. The situation may have shifted already. So it sounded very, very complicated. This was the PhD student who worked on this. So we just decided, let's go for a genetic algorithm, and always trying to make things compete with each other when the S and the M values for every week in the system can dynamically change. And we just sort of competed the hell out of the situation until the solutions started converging. But they converged quite nicely. And
sorry, can I ask a question?
Yes, please.
What is it that the choice can condition on? You said the choice whether to produce. Does it condition on like temperature or counting time?
So I will come to this at the end of my talk. What it looks like empirically, but I can tell you what we did in the model. In the model, we assumed, and here I should say that if we had had more time, we could have probably also explored other options as well. In the model, we simply assumed that they just know that now it's week 15 of the summer, and this is what you see on the x-axis here, and that they also, evolutionary speaking, they also know that everybody else is behaving in a particular way, because this is repeatably the condition that the pond is this full and all that. But they don't actually actively measure in our model how many males they are encountering or whatever. They just know that, okay, now it's week 20 or it's week 25. Okay, so what the algorithm is doing, it's seeking a combination of S and M values for each week separately that produces the best geometric mean for the number of sexual eggs accumulated by the time that the season ends. And we use the geometric mean because these are discrete seasons. And if you have learned your bed hedging theory in this sort of discrete setting, evolution will actually optimize the geometric mean, not the arithmetic mean. This is what the population genetic results point you to do when we have this sort of clear cutoff from one season to the next. And what you see in these graphs here is, okay, so we have the week number starting from the first possible week when reproduction is possible. And the population density increases, and sometimes you can see that it varies. And the reason why it varies is that if the population has started to produce these sexual eggs, they just go to the bottom, they wait for the next spring, so they don't contribute to the current population size at all. Whereas if you go for directly developing offspring, then of course that increases the population. There's also a certain mortality rate, so that, you know, this is kind of like the net increase that you see in the system. And what you see here in these sort of vertical lines, this is the probability that the season has ended by this time. So here we have a very sharp end of the season, and everybody, evolutionarily speaking, knows that they have a lot of time. They have 30 weeks before everything ends. Whereas here there's a lot more uncertainty about when it might end. And here already from week 10 onwards, there's some chance that it may have ended, and for sure it's ended by week 30. And you can see that this has big impacts on what they should be doing. And very interestingly, you know, well, it's kind of obvious that the model should produce that male production, which is the pale gray here. It always starts 2 weeks before sexual egg production, so that the males, it's pointless to produce sexual eggs if nobody has been producing the males. And because it takes 2 weeks for the males to develop, this is a kind of like a very nice way to schedule the system. But you can see that both when there's very little uncertainty and also when there's a lot of uncertainty, you end up in a situation where you always do a little bit of everything late in the season. But you can also get these situations where you go through these cycles where sometimes you go for sex only, then the population starts declining. Then you start with, you know, filling the pond again with your females. Then you start going for sex again, and so on. So you can get into these sort of interesting alternations of what is happening there. And for some reason, I took out the slide, but we also wanted to see if this really does satisfy the formal definitions of bed hedging, that if they did go for just the arithmetic maximization of everything, then the variances and the means, they behave in a way that satisfies the definition of bed hedging, and it does. So I hope you sort of buy the story in the sense that, you know, this is actually one of those quite curious cases where evolution really does operate through bed hedging instead of just sort of trying to maximize the mean offspring production. But there was already a question from the audience, like, you know, what do they actually know about their environment? And in the model, we just sort of fairly simplistically just thought that, okay, you know, they have had thousands of years of experience of the system. So therefore the scheduling works in a way that if a Daphnia knows that it's week 30, then they kind of like know that it's time to switch to a particular kind of behavior. But in reality, of course, you know, they don't have clocks inside them. They don't really have calendars. They probably must be using some cues about what the weather is doing, or how many others there are, or things like that. And people have earlier already looked at things like, you know, the day length, producing some really quite interesting results, because Daphnia seem to respond to that differently if they're from the very north or if they're from the south. Like in Central Europe, where the drying might be a bigger problem than the winter coming very soon. It's actually the longest days that cause a lot of sex to happen. Whereas in Finland, where it's really kind of like the very, you know, the drying is probably happening a bit less often, and the winter is really the hard thing at the end, the responses are a bit different. But we also wanted to know that they might use their biotic interactions with others as a cue for deciding how late it might be in the season. Because if you remember, everybody starts out as a female, and also in the beginning of the season, the population is at its lowest because not everybody survives the winter, and only some of these hatch, and initially there's not so many Daphnia in there, and certainly there's no males. Males appear gradually. So this is what we wanted to experimentally test, if we can trick the Daphnia to believe that it's earlier or later in the season. So the weather conditions are the same for everyone. We even randomized the order of the buckets so that if the sun is shining from a particular angle, then we have different treatments on kind of like every one of these rows, and buckets are white so that they wouldn't warm up too badly, hopefully. But we manipulated the density and the sex ratio. So in two different ways. So one is that the different buckets had no males or some males or many males in such a way that the density simultaneously increased if we added more males. But also we did the same thing so that we kept the densities constant. So here, the idea was that we can independently look at, do they respond to each other's densities? Or do they also make a difference between whether the individuals you're bumping into are male or female? So what we found, and this is actually, this is quite astonishing, experimental biology doesn't always work so well. But we ran this experiment 3 times with slightly different settings, and we got these are estimated response curves of how much male production occurs if you have started the buckets with a lot of females or a lot of males or a combination thereof. And look, these are all independent experiments, but the response surfaces are incredibly repeatable. So it feels like, they really do behave very consistently in these settings. So if you add density to a population so that the additional individuals are female, what happens is that they start producing more males, which is logical, because this seems to signal to them that, okay, population has been increasing, it must be quite late, so therefore I might be a male producer now, because late means that sex is about to happen. But remember, we could also do this sort of increase by adding males to the situation. So then it gets a bit more complicated. What would you even predict? Because on the one hand, if there's males in the system, that means that it's probably late in the season. But on the other hand, if I now, as a mother, produce a lot of males, it means that they are competing with males who are already in the system, except that my sons would only mature 2 weeks from now. So what are the chances that these existing males are still there? And that's why we needed to do a genetic algorithm in the first place, because it was sort of complicated to work through what is even the ideal behavior. But maybe if you're thinking through with me, you would say that probably you should increase male production, but maybe not as much as in the first case, where everybody was a female who is likely to produce some eggs that need to be fertilized. And that's actually precisely what happens in here, so that you can see that the male production is increasing in this direction as well. So this is a sort of positive slope, but it's a much milder positive slope than here. It's only about 40% as steep. So male production does increase if there's a lot of males in the system, but only by about 40% as much as if there's a lot of females in the system. So I felt that this is really quite exciting, and I'm quite pleased that it worked out so beautifully. And I thought that I'll just end here already. I didn't want to make this talk like super long, so that there's time for questions. The take-home messages being, well, remember the first story where I was talking about all these sort of imaginary organisms. Your theoretician friend has not necessarily gone crazy, even if one wants to look at some completely hypothetical alternative scenarios. And remember that these really unusual cases, they also do exist in nature, and it can be really, really fun to think through what is actually the best way to act if the rules of life are quite different from what they are in situations where, you know, maybe we as humans are more used to thinking what should happen there. And timing traits as a whole, I think, for example, Christoph knows this, that I can also talk about those kind of things a lot. For example, the germination problem that I mentioned briefly, we also have a paper on that, and the migrating birds and all those kind of things. When should they even come back? And but I don't want to make this talk too long, so I just thought that I'll end here, and I will thank all the co-authors of these particular papers. And yeah, thanks for listening, and thanks to all the institutions that funded this study. So I'm done here.